Hello friends, how are you all? And welcome to my channel Delta Pharmacy Classes, and I hope you all are fine. So today we will discuss about the structure of bacteria, and before this, first we will discuss about the type of bacteria. We know primarily bacteria having three type of structures. Number first is spherical, then these bacteria is called coccus. And if bacteria have a rod-like structure, then these type of bacteria is called as bacillus. And if the bacteria have structure like this, then what these called, that is called spirochete. And these type of bacteria or also have subtypes, like if they have one spherical like structure, then these called monococcus. And if they have two spherical structures, then they called diococcus. And if they have a channel-like structure of many spherical structures, small structures, is called streptococcus. Friends, one more type of bacteria which don't have a regular shape, which not looks like real bacteria, but actually they are bacteria. Then what are the name of this type of bacteria? And they have a structure like and they have only cell wall and genetic material. And these bacteria are the smallest bacteria which looks like as virus. So what are the name of these type of bacteria? So these type of bacteria are called mycoplasmas. Now friends, today we will discuss about the structure of bacteria. So today we will understand that what are the regions that the antibiotics are not responsible to kill the bacteria, and how they stop the growth of bacteria. So today we will discuss about this. So we know, like this is the structure of a cell, bacterial cell. And this membrane is made from a phospholipids bilayer, and this is called cell membrane. This is called cell membrane. All its organs, the cell have many cell organelles and components. So these all components are inside this cell wall. So this membrane is also called as cytoplasmic membrane. Yes. And what friends is there in this structure? There is also one layer outside this membrane, and what this membrane called? It is called cell wall. So friend, how it made? So we will discuss about it. These structures are called backbone of sugar. What called? Backbone of sugar. And this is also called as we can say it's peptide. What? Oh, glycans, also called as glycans. What? Glycans. And there are some small structures in these structures, on these structures, and these small structures called peptides. What? These are called peptides. What call this? Peptide. And there's a connection between these pipes, between these links, and this these are the links between the peptides, and glycans are connected each other and forming a structure called peptidoglycans. We know that the cell membrane, the cell wall made from peptidoglycans. So this is the structure that made from glycans and peptide, and this layer is called cell wall, which is made from peptidoglycan, and this is the between these peptides there's a linkage between this, so this is also called as peptidoglycan linkage. So I'm just think that this structure which is inside this outside the cell membrane, so this is one molecule or having a lot of many molecules of peptidoglycans. So friends, this is not, there is not a number of peptidoglycan. This is the mega macro molecule of peptidoglycan, which is made from sugar backbone and peptides. So it is only the one single macro molecule of peptidoglycans, and we know that inside the cell of bacteria there are some organelles, and inside this there is a fluid inside this. This is called hyperosmotic fluid. And when this virus enters in our body, so we know that there are so many fluid inside this, that is called like inside us there are many like sputum or saliva. So when they comes in contact of saliva or sputum, so what happens there, we know that our body have a hypoosmotic fluid. So what happens there, according to me, the liquid, when it enters in our body, so the fluid of our body should enter inside this. What? These peptidoglycans do not allow to enter the fluid through this, because this is not a barrier, and it not help in transfer of materials or fluid from inside to outside of the cell. So and this is not a barrier, and cell membrane is a good barrier, and it is very very porous, and so the cell membrane is responsible for the transportation of nutrients and fluid inside this. And what are the main function of cell wall? So we know that it gives a particular shape to the bacterial cell, and second one is the most important factor is that it helps in protecting from when a fluid in start to inside this start to enters into this through the osmosis process. So friend, the size of cell it just start to swell. So this cell wall have a main function is that it is not allowed to swallow become this cell of bacteria. So it helps to maintain the shape and size of the cell. So during when bacteria enters in our body and fluid is start to enters into this and cell size of bacteria is start to large because fluid is start to enters into this. So this cell wall does not allow to swell this cell and hence prevent it from bursting, you know. So this is the main cause that the antibiotics like penicillin, when we consume penicillin vaccines, and what happens, we know that there is hyperosmotic fluid, and when we use antibiotics, they are not capable to disrupt this cell wall. They are not capable to disrupt this cell wall, and hence antibiotics are not capable to destroy the bacteria. But actually the main working of antibiotics is that they reduce the growth or they stop the growth of new bacteria. What are the reasons when friends, now we will erase this, and now then we will understand that how it happens. This is the parent cell, this is parent cell, and these are two daughter cell. These are two this is also daughter cell. So when they start to form new daughter cell, then the fluid inside this is not hyperosmotic, this is hypoosmotic, and their cell wall, the cell wall outside the cell membrane is not fully formed. Whenever outside this there is a cell wall, so when they start to grow, then the fluid inside the daughter cell it is start to convert into hyperosmotic. Its concentration is start to increase. So there is also start to form cell wall. So when the fluid inside daughter cell is hyperosmotic, then the cell wall is not outside this cell. So this is the main reason that when we eat antibiotic, then it is not capable to disrupt this cell wall. It capable to disrupt their growth like that. When they form daughter cells, then when we consume antibiotics, then these antibiotics does not allow to form this cell wall, and due to which their fluid inside these cells is becomes hyperosmotic, but their cell wall is not fully formed. So antibiotics start to disrupt this cell membrane and stop the growth or formation of new cell wall. So this is the reasons that the antibiotics which we consume are not responsible for disrupting the cell wall of the parent cell, means bacteria. Actually they stop the growth of new cells which are forming through these parent bacteria. And we know that if there is one or two bacteria which enters in our body, does not are not capable to harm you, because one two bacteria are not capable to harm you when there's so many of bacteria, or then they start to form many daughter cells and forming a lot of bacteria, then they cause disease inside the body. So friends, this is the overall concept about the cell membrane, cell wall of bacteria. And in next lectures we already have studied about the nutritional requirement of bacteria, and now next we will study about the pure culture medium. Isolation of pure culture medium is our next topic. And thank you so much for watching, and keep